So how do you actually cure a genetic disease if you don't know exactly where the mutated gene is even doing its damage? Well, welcome to our virtual laboratory. In this explainer, we're embarking on what's basically a molecular detective story. We're going to uncover exactly how researchers map precise protein binding sites right on our DNA in order to cure severe blood disorders, things like sickle cell disease and beta thalassemia, that affect literally millions of people across global communities. So let's dive right in and decode some of the molecular secrets hidden inside our cells.
I want you to just think about that initial problem for a second. Our DNA is incredibly vast, right? And gene expression is orchestrated by thousands of transcription factors. So how can researchers possibly visualize protein DNA interactions across the entire genome to solve the mystery of fetal hemoglobin silencing? I mean, the logic here is simple. If we want to cure genetic blood diseases and deliver real relief to our communities, we have to know exactly where the culprit is operating.
And the answer to that massive question is a revolutionary technique called ChIP-seq, which stands for chromatin immunoprecipitation sequencing. Now, you can think of ChIP-seq as a kind of molecular GPS tracker for our DNA. It profiles transcription factor binding events across the entire genome, answering that one absolutely crucial question: where exactly does a specific protein, in our case, the master regulator BCL11A, physically attach to our DNA to regulate genes in living cells? To get there, here's our roadmap for this explainer. First, the ChIP-seq workflow. Second, the research logic pipeline. Third, BCL11A binding and cut and run. And finally, why Indian cohorts matter. Okay, let's kick things off with part 1, the ChIP-seq workflow. So, how do we actually catch a protein in the act? Step 1 is cross-linking. We add formaldehyde to the cells. Why do we do that? Well, it essentially freezes the scene of the crime, locking the DNA and proteins together exactly as they are in a living cell. Next up, step 2, is chromatin fragmentation, where we use sound waves or enzymes to break the chromatin down into tiny 200 to 500 base pair fragments. Then we hit step 3, the immunoprecipitation part. This is where we add an antibody that's highly specific to our target protein, like BCL11A, and then use magnetic beads acting like tiny molecular magnets to pull down the BCL11A along with its attached DNA. And finally, step 4 is a thorough wash just to get rid of any non-specific DNA or contaminants. Okay, so once we've isolated our target, we move to the second half of the workflow. Step 5 is reverse cross-linking. We use heat to melt that freeze we created earlier, breaking the bonds and releasing the pure DNA. Step 6 and 7 cover DNA purification and library preparation. Then step 8 is sequencing, which generates literally millions of DNA reads. And step 9 is where all this hard work pays off: bioinformatics. The software sifts through all those millions of reads to find what we call peaks. And these peaks, they are the enriched regions that prove exactly where our protein, BCL11A, was bound.
Which brings us to part 2, the research logic pipeline.
This pipeline brilliantly illustrates the scientific method in action. It's the complete genetics workup, so follow the logic here. First, GWAS, genome-wide association studies, discovered that variants in an erythroid-specific enhancer actually control BCL11A expression. And this explains about 15% of fetal hemoglobin variation. So GWAS basically said, hey, this enhancer exists. Second, ChIP-seq stepped in to confirm the direct binding mechanism, showing us exactly where BCL11A attaches. And finally, RNA-seq provided the functional proof. It showed that when you knock down BCL11A, fetal hemoglobin levels increase dramatically. Discover, confirm, prove. It's multiple methods, each perfectly validating the other.
Let's move to part 3, BCL11A binding and cut and run. Now, we have to ask, does BCL11A work alone from a distance, or is it operating directly at the scene of the crime? Well, high-resolution analysis revealed a pretty huge surprise. BCL11A doesn't just bind locally. It has long-range interactions at the upstream locus control region and intergenic regions, which actually reconfigures the chromosomal loops of the beta-globin cluster. But crucially, and this is the absolute key to everything, it directly binds to the gamma-globin promoter. That direct repression right there is what silences fetal hemoglobin in adults.
So the crucial point for researchers was finding out exactly where it binds at that promoter. And they found that BCL11A targets a highly specific 6-letter code, TGACCA. It's kind of mind-blowing to think that such a massive life-altering disease comes down to just a few letters in our genetic code, isn't it? In the human gamma-globin gene promoter, there are actually two copies of this motif, located at positions -119 to -113 and -92 to -86 relative to the transcription start site. We literally caught the culprit exactly where it operates.
But BCL11A doesn't act alone. It has co-conspirators. BCL11A physically and functionally interacts with another transcription factor called SOX6. Together, they co-occupy the beta-globin cluster and cooperate to silence gamma-globin. But here's where the molecular drama happens. They are in a fierce competition with an activator protein called GATA1. When BCL11A and SOX6 bind, they repress fetal hemoglobin, but when GATA1 wins out, it activates it. So, this tiny molecular tug-of-war determines exactly which type of hemoglobin your body is going to produce. So, let's throw a new question out there to test our logic. If BCL11A binding causes the silencing of fetal hemoglobin, what do you think occurs in patients where that specific binding site is naturally broken? The answer to that provides the ultimate proof of our mechanism. You see, there's a naturally occurring condition called hereditary persistence of fetal hemoglobin, or HPFH for short. In these individuals, single-base substitutions at positions -117 and -114, or microdeletions in that exact promoter region we talked about, physically block BCL11A from binding. And the result? Fetal hemoglobin levels shoot way up, and these individuals remain perfectly healthy. This natural mutation gave scientists the exact blueprint for a cure. The logic is undeniable. If we can stop BCL11A from binding, the silencing fails. But as with every good scientific detective story, there's a plot twist. Despite how powerful ChIP-seq is, numerous attempts to localize the naturally occurring endogenous BCL11A actually proved unsuccessful. The protein was incredibly difficult to detect. So how did scientists finally manage to map it? Well, I had to upgrade their tools. Researchers adapted a newer method called cut and run, which stands for cleavage under targets and release using nuclease. So why did it succeed where standard ChIP-seq struggled? It's because cut and run requires no formaldehyde cross-linking. By skipping that harsh chemical step, it preserves the delicate native state of the chromatin, offering much higher resolution and detecting protein occupancy directly in the chromatin itself. Because of this enhanced sensitivity, cut and run was finally able to detect BCL11A occupancy in those gamma-globin promoters. It just goes to show, sometimes advancing the research logic requires advancing the technology first. Which brings us to our final section, why Indian cohorts matter. Let's bring this all back to the communities impacted. Sickle cell disease and beta thalassemia affect millions of people in India, especially within tribal communities in states like Maharashtra, Madhya Pradesh, Gujarat, Odisha, and Chhattisgarh. Now, the genomic logic we've discussed today makes CRISPR and base editing of the BCL11A enhancer a massive potential cure. However, we are facing a severe genetic diversity gap. Most GWAS data out there is based on European or East Asian populations, but Indian populations have unique genetic diversity and potentially completely different BCL11A enhancer variants. If we don't study Indian populations specifically, the gene therapies built on European data just might not work for Indian patients. Which leaves us with this final, absolutely vital question. Will the next big gene therapy be built for your community's DNA? We urgently need localized genetic research, cohort-specific genomic data, and targeted analysis so that these highly advanced CRISPR cures actually work for everyone, not just a select few. I invite you to join this mission to ensure that the genetic workup we've explored today translates into real, accessible cures for communities across the entire globe.